Welcome to the Proteomics course.
All we need to find out the fastest sequence for given protein so that you can search many of the databases for this purpose. Most commonly used database can be UniProt database, so here you just have to give the name of the protein and then you can download the FASTA file. And then you can import that FASTA file in the Skyline software. So this is the screenshot of Skyline software. So once that FASTA file is imported, then you can see the list of peptides it has generated. You can actually give the filtration criteria for this generated list. This is peptide selection criteria. Here you can specify which enzyme you are using for digestion. You will be selecting Ms cleavage as 0, to get better, accurate results. Then you can select peptide length. What should be the ideal length of protein for triple quadrupole system? Most commonly it will be 825 amino acids, and if modification is tend to happen in your peptide, then that modification you can specify like oxidation, phosphorylation. If you are using heavy labeled peptide, then you can specify that as well in peptide selection criteria. Once the peptide selection criteria are set, then go for transition selection criteria. So in that case you will select what should be the charge on precursor MZ, what should be the charge on your product MZ. You will select which type of ions you want to monitor, because when peptides get fragmented there are A, B, C and X, Y, Z, all these 6 types of ions will form. But out of these, Y and B ions are most predominant. In that Y and B also, Y are most predominant. So in majority of cases, you will prefer to go with only Y type of ions. Then you will select what should be the length of product ion that should be considered during the fragmentation. So you can specify here ion 3 to last ion -1 or ion 2 last ion -2, depending upon your requirement. Once that is set, you can see the list of transitions generated like this. So you can see here almost 160 transitions generated for selected protein. After this you will just export this transition list. Here you will just select the Shimadzu instrument, because if you are performing your analysis using Shimadzu triple quadrupole instrument, you have to specify the Shimadzu instrument. In one method you will be exporting all these transitions. Once those transitions are exported, you will open your lab solutions software, which is nothing but acquisition software for Shimadzu's triple quadrupole instrument. In that solution software, you will import all these transitions. After that you will specify what should be the time program, HPLC time program, whether you want to do staggering, that staggering you can specify, what column you want to use, that you can specify that. After all these parameters optimization, you can give a run and perform the analysis. After the analysis is over, you can import that result or data file in Skyline software again, and you will be seeing here different colored symbols. For a given peptide, if all the transitions which are generated by Skyline show positive results, then that peptide will appear in green color. Suppose if any of the transitions is not generated any response but remaining others are generating response, then that peptide will appear yellow in color. If none of the peptides has generated the response, then that peptide will give red signal. So depending upon your selection criteria, you will select which peptides have to be kept for further analysis and which have to be removed. So most of the peptides which are giving you red color symbol, you will refine them in the filtration, and you will keep only those peptides which are giving green color symbol. And then those transitions you can use for further analysis. On that method, you will be analyzing all of your samples, and then you can compare the peak areas for all the samples and perform the relative quantification analysis. Now let us see how to do this thing practically. So this is blank skyline document. Now we want to find the FASTA sequence for protein called as vimentin that we can get from UniProt site. This FASTA sequence you will copy and you will paste it into Skyline file. Once the FASTA sequence is pasted in your Skyline file, you will see a huge number of peptide sequence and transition list generated by Skyline. Now you can use some refinement or filtration criteria. You will go to Setting, change Peptide Setting. You will specify which enzyme you have used for digestion. Maximum missed cleavages will be 0. In Filter, you will specify what should be the length of the peptide. It will be generally 825. You want to exclude potentially ragged ends, and if you want to exclude peptide containing some special amino acid, you can just click on that amino acid here. In that case you will select precursor and product ion mass as monoisotopic. System will be Shimizu triple quad system. Collision energy is already optimized for Skyline and Shimizu triple quad system by Shimizu Research and Development Center. Depending upon the transition, it will tell you what will be the best optimized energy. In filter criteria you can specify charges on precursor ion, 2, 3, you can specify charges on product ions, and one can select which type of ion you will be using for quantification. Most commonly used is Y type of ion. Then you can specify the length of the product ion which needs to be considered during the fragmentation, which is ion 2 last ion -2. In case if you want to mention some specific modification, you can just check on that modification. Now you will save this file and you can export the transition list. Now you will go to Lab Solutions software, which is Shimizu's acquisition software for triple quadrupole instrument. Once analysis is done, now you will open those data files, which you have analyzed in your Vimetine Skyline file. For that you will go to File, Import, Results. You can see results have been reported in the Skyline file for both these data files. So, you can just adjust the data files, and here you can see in the remaining peptides there is no response generated. Only for these 3 peptides there is response generated. But still they are looking red because for some of the transitions, there will not be any response. So here only for these 2 transitions response is there, and here also for 3 transitions response is there. And in this case also there are many transitions for which response is generated. Now you want to refine these results. So, you will go to Edit, Refine, Remove Missing Results. All the missing results are disappeared, and only those transitions are the for which response is generated in triple quad. So you can see the peak shapes for both these data files, and you can scroll down to check how peaks are looking. If you see for a given peptide, all the transitions are properly coeluting at the same time retention time, its peak shape is also good. And if you want to compare the peak areas, you can just have this graph. Both of these samples belong to same grade of cancer, so their area counts are almost similar. You can actually compare peak areas for different grades of cancer and see if area counts are increasing or decreasing, and you can check what is the relative expression of given protein in case of cancer samples.